I'm Bhavidhir V. Bharadwaj. Welcome to episode 35 of Genesis of Tomorrow. The following is going to be a conversation about bioinformatics, genetic and molecular biology, quantum strains and explanations, and much more with Dr. Rowan Beck, an esteemed scientist and scientific communicator. Also, folks, if you're interested in such deep science and deep tech conversations and are able to garner even the slightest bit of value from the show, please support us by clicking the subscribe button so that we can keep improving the quality of the episodes and keep on bringing such wonderful guests over for you. And now, without any further ado, let's get right into it. Enjoy and thank you. And we're rolling now. Okay, so first of all, a formal welcome on the show, Dr. Beck. It's
Thank you for
lovely
having
to
me.
have you here, and I'm really excited to see where this conversation goes.
Thank you for having me. Yeah, I'm excited. I've never done anything like this before, so
Okay, so I like to do this with a lot of the topics, with a lot of the guests that I interview, which is primarily asking them to lay a foundational basis of their said subject matter. In your
mhm,
case, anything and everything to do with bioinformatics and ancillary topics.
sure. Yeah. I can give some background if you're interested, just a little bit about myself. So I do have a
Certainly
background
so, yes.
in. I was going to say, sorry. I do have a background in science, particularly with a passion for communication. So, as you mentioned, bioinformatics. I received a PhD in genetics and molecular biology. Sorry, from UNC at Chapel Hill. Um, And then before that, I had a BS in biology from the University of the District of Columbia. So my research has kind of been all over the place. It spans, scientific research in toxicology, bioinformatics, gene regulatory systems, and I've worked within regulatory affairs, genomics, and bioinformatics as well. And yeah, I am particularly excited by science communication as well as mentoring and trying to educate the next generation of scientists.
I think a very significant or seminal aspect of the entire any field at large is why is it so seminal in its endeavor for study or advancement. And even though it's very self explanatory with a field like yours, but still, like, in what all different magnificent ways can bioinformatics and, even other things as well, you talked about molecular and gene biology,
Mhm.
how all of these things can be just downright interesting to somebody.
I particularly love bioinformatics, let's say specifically, because I think it kind of is the intersection of computing, mathematics, biology, human health. Um, Understanding bioinformatics really it takes us to the future. So without the bioinformatics, how can we understand ourselves? How can we treat disease? How can we understand differences between people? Um, yeah, I don't know. It's the intersection of everything I like, so it makes sense that I chose to study it. Like I said,
Even though it's a multidisciplinary sort of amalgamation of all these different fields, has the growing prevalence of artificial intelligence at large affected its usage, but more so, during the research end of the spectrum? Like, has it affected that in any way?
it's growing. I absolutely do think that the inclusion of AI, machine learning algorithms, any kind of advanced computation has influenced bioinformatics and biology as a whole. Whether it's drug discovery and simulations done on, just massive computational servers, or trying to predict protein structures and foldings and things like that. I'm using a lot of AI and machine learning in particular for assessing images, actually. So predicting whether an image falls into a certain category or not, whether it's cancerous or not, based on previous training. So I do think it's still early on, but it is growing. Everything we discover using AI, machine learning, whatever, still does need to be validated in the laboratory. And I think that's kind of the holdup. A lot of it is theoretical. A lot of it is, it's great for a particular data set, but does that mean we can apply it to a larger data set? We're still trying to find out to validate everything.
that also sort of in some way paves a more quicker road in terms of reaching a state in personalized medicine and
Absolutely.
personalized healthcare. But like, how exactly do you figure out that perfect balance while you're working with so much of data, which for the most part might be initially a lot more messier than usual, and you'll have to go through the entire process of pre-processing it, cleaning it, and then training your models and everything. Because I really do not know how it all works in the biological field.
Hmm.
How exactly do you work towards formulating or advancing a personalized approach to medicine?
That's a great question. I think the first step, regardless of whether you are incorporating AI or quantum computing or whatever else in your workflow, is trying to consolidate the various data sets and have a consensus on one, what data we're collecting, what format it's being collected in, what format of just what file types, what associated data. I think that's actually one of the largest holdups when it comes to analyzing data. Obviously, coming up with your scripts and your workflows, et cetera, is challenging. And that's the core of bioinformatics. But I think a significant portion of that time is spent actually just cleaning the data and trying to reach a consensus among different consortia and groups so that the data makes sense when you combine it, because there's no sense in having data that just it doesn't make sense when you put it together. So I think that's a pro and a con. It's great that we have various institutes and consortia and hospitals, whatever else, collecting data anywhere they can, as well as in the laboratory generating data, doing experiments. But we do need to find a good way, and potentially using some kind of AI or something to combine this data so it actually is constructive instead of just kind of interfering with each other.
You also briefly mentioned that you had some interest in the regulatory process of side of things, right?
Mhm.
From just a pure perspective of curiosity, how does all this technical research work that you conduct in your labs and everything, how does that translate towards the broader implication for other people in the field, maybe doctors or maybe hospital workers, to actually facilitate that new state of the art technology or research into their day to day procedures?
Yeah, great question. I think that's always a challenge. You could have the latest, greatest, most state of the art technology or algorithm or whatever it is that you are working on, but if it's not been validated over and over and trusted, it's very difficult to get anyone to actually adopt this methodology, especially when working in regulatory affairs, working with government institutes like the FDA or just any NIH. Rightfully so. Everything needs validation. It needs considerable testing on a variety of test subjects, whether it's in the lab or it's like clinical trials, and you need a diverse set of individuals to test on. So I think it is slow moving, but for a reason. I think just again validation of everything that is being discovered. But more and more people have access to a lot of these workflows, these state of the art tools, algorithms, etc. They can try things on their own, whether they've cloned something off of GitHub or just they've stumbled upon some AWS workflow that seems to work really well for them. So more and more people are adopting these types of technologies, I think. But I do think it will take a bit of time before federal institutes or any kind of government or regulatory kind of institution is willing to adopt it in a timely manner.
Then how do you really try to tread that line of balance between not being too late at inculcating a new
Hmm.
way of doing things, especially in a field that has so high of stakes, right? So you're
Yeah.
not as late into adopting that new approach while also making sure that it is decently, in a well defined manner, tested and worked through thoroughly.
I think the ideal answer is to do both, right? Use a known methodology and then, pair it with something new to prove that they say the same thing, that you get the same results. That is not feasible for many companies, many organizations, of course, which is, I think, what is problematic in some of our healthcare. We have these really big players who can afford to spend tons of money, reproducing their workflows and doing multiple methods to validate, which is great, fantastic. But it does leave out a lot of the smaller players who have to either just take a leap of faith to use the newer technologies, and that's where you get these startups that just appear out of nowhere, or stick to what is known and accepted, an old assay that might not be the most effective, but we know it works pretty reliably. Um, so I think that's the answer: is just do the new thing and validate it with something that we have established already, if you can.
And where exactly do we stand in terms of what we are capable of in doing in the medical field? Like, how advanced are we? What all can we do right now? So as to better understand what all we are working towards in the future.
Yeah, that's a great question. We can we can do quite a lot, but it depends on how able we are to scale that. Personalized medicine, I think, is easy for one person, but for an entire population, it's difficult. There are still things we don't even understand about why certain populations are predisposed to certain illnesses, or even just current drug discovery. It's being tested on essentially men rather than women, but then that doesn't actually answer any questions about why it interacts differently with women. So we can do this. I think it is costly, and I think it would produce data that doesn't necessarily always align with the data that exists. And I think that is where it complicates matters. We can only, solve a small puzzle at a time. And you'd think the more pieces, the better the picture, which is true. But you really just need to slowly collect all these pieces over time to paint the picture, to actually put it together and make it make sense. Because until you have all of the pieces, it just looks like, three separate puzzles that you're working on at once.
Okay, I might seem from here way on a little bit out all over the place because there are just a lot of different matters of intrigue that I wanted to pose questions about to you. But starting from maybe the entire thought trail of tinkering with genetics, right? Genetic
Mhm,
modification, enhancement, gene editing, molecular engineering, all of that.
mhm.
What is the entire domain about? And
Um,
what is its potential as such as well, like to its highest cadre?
well, if you ask an RNA fan like myself, it's endless potential. But then, you talk to someone who goes further down the line with proteins and things like that, they'll tell you that's where the future is. But I think it's both. Just to catch everyone up, generally you have DNA, which gets transcribed into RNA, and that's kind of the active portion in the cell that's currently coding for genes. And then those genes act through proteins. So which part makes sense to study? All of it, honestly, because without the DNA folding and changing its structure, you wouldn't get the RNA, you wouldn't get the proteins. Proteins might be carrying out the orders of the DNA, but it's all intertwined. So that's why we really need to develop all three kind of technologies to study all three aspects at once. Um, but just speaking about gene expression in particular, like we're just discovering new types of RNA every other week. I went to school. It was messenger RNA. We had ribosomal RNA. I studied a lot of microRNAs, but there are circular RNAs that act as sponges, tRNA derived RNAs. Like, it's insane the amount of new molecules we're discovering and trying to understand then, are they real? Is it an artifact? And we have nothing to base that off of. But we do know that they interact with one another. So, we're trying to understand that and we're trying to study that to really unlock the potential of gene editing, gene expression, because you can change a gene, but how do we know that's going to have long-term consequences? And I mentioned I particularly I'm a fan of RNA. I love studying RNA. I think it computationally makes a lot of sense to study RNA. Proteins fascinate me, but for each RNA, you know, that codes for a protein, it can code for multiple proteins depending on how the protein folds. So there's an infinite possibility with each, strand of RNA that it will create something new. Um, Studying those new folds, those proteins, that's computationally very costly. It's not always the most efficient, and honestly, we don't have the best softwares for it, which is why it's a lot of it's just a little bit slower. That's why there's a lot of focus on gene editing, kind of changing the protein from its source. I do think there's a lot of potential there, but I don't think we fully understand the implications long term, which is what is slightly worrisome. And yeah, I don't know. It excites me. I really like it. I think it's great that we are trying to tinker with things like this. But relating back to my earlier points, everything does need to be validated in cells, then moved to animal models, then whatever it is long term. And half the challenge is finding something that then represents what it would be like in humans. We can study mice all we want, but they're not necessarily the same. It's a great model organism for some aspects, but what does that mean for humans? And I think that's the challenge. I feel like I've rambled. I apologize.
No, 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 I think it was pretty good. But maybe a more of a scientific breakdown for people who do not have as much of a thorough technical background in the field. How does this central dogma of sorts of molecular biology, the DNA, RNA, protein interactions, and all those things, how do they actually will work in terms of what we have to study in them to reach a point in making meaningful bio enhancements or genetic enhancements?
Yeah, I think that's the $1 million question. That's challenging to answer because there are so many different directions you can study these aspects. I mentioned essentially you start with DNA, you have RNA, you have protein. So each element can be studied through multiple lenses, multiple technologies. So it's really hard to try to predict where we are in these, and what we can do with everything. I mean, you could solve all the riddles you want in just RNA, for instance, and then find out that none of them actually translate or mean anything for protein folding. Or not only that, but you know, chromatin accessibility can alter what RNA is expressed and therefore alter whatever gene expression you've modified or interacted with. I think it's really complicated. I don't know. It's There are just so many different angles for each of the components. I do think obviously there are people studying each of these aspects. We are trying to pair a lot of like just for example, gene expression now with imaging analysis so that you can actually find out which genes are expressed where in a tissue, which is fascinating. Previously, kind of, we'd look at RNA as just a whole, a bulk. So it was kind of like you would take a tissue and crush up all of those cells and get the RNA, and it would tell you an average across all different cell types, which doesn't really necessarily tell much. It just tells you in that tissue, you have more of this gene, but it doesn't tell you where that gene is located, what types of cells are expressing that gene, what it means long term. So more and more of the technologies now are moving toward single cell sequencing, is what it's called. So it kind of separates out all of the cells in a given tissue, and from there you can look at the different gene expression among all of the cells compared to one another. And I think things like that, really looking at a fine micro scale of everything, will help us to understand diseases and their impacts, because previously you would have no idea where to target that there was a gene that was abnormally expressed, or you know um, if you were just looking at the tissue as a whole. But now you could say that, in this group of cells within the heart tissue or whatever, within the lung, this group of cells is expressing an unusually high amount of this cancerous related gene, right? So now we know where to target it, we know what to look for. So, and not only that, but comparing that with then a medical image of the lung, a scan, you can actually identify where those genes, where those cells are within that lung. So you could really identify highlight expression and then know where to target it. So I think in that aspect, targeted therapies and precision medicine is where it's heading. Just we are combining a lot of existing technologies together to try to understand how they're interacting and get a finer picture.
As a comparative analysis of sorts, with a with with a disease like maybe the coronavirus, and then on
Mhm.
the other hand something like cancer, why is it that making a cure of vaccine, particularly of something like coronavirus, was way more comparatively easier than making or finding a cure to something like cancer?
I think the answer to that is we have some experience with viruses. We know generally how they operate, which is they have their own set of kind of genetic material, and they invade our cells and get us to release antibodies and things like that. I think with the coronavirus in particular, we were quick. We were quick to sequence its genetic material and find out what it was carrying, and from there, kind of give a heads up to the body, right, through a vaccine. That's kind of what they do. The mRNA vaccine just gives a clue to your immune system of what to look for when you are exposed to something. Cancer is more complex in that there are many different thoughts of kind of how it starts and the different genes, the different causes for it, I guess you could say, the different molecular mechanisms underlying the different cancer types. Even the same cancer type can be some underlying differences in molecular mechanisms. And I think that is the problem. Is trying to find one solution is not going to solve it. When people say like, let's fight cancer, let's cure cancer, it's not possible because which cancer? They all kind of work differently in the body. So I think that's why you see a lot of splits between the different types of cancer, breast cancer, prostate cancer, because you really have to study the tissue of action as well as what's happening within that tissue. Different tissues respond differently. They respond differently to treatments. So that's part of the reason as well that you can get treated with certain medicines. But you end up damaging the tissue more than the cancerous part of the tissue, and that's what's dangerous. So it's kind of finding a fine line of what you can target exactly without hurting the person. With vaccines and viruses, it's less of a fine line. We kind of know what to expect. But even with the coronavirus vaccine, as your example, some people did react. They responded pretty negatively, adversely to that vaccine. It caused a lot of inflammatory response and things like that. So even with that vaccine, it wasn't perfect, but we knew kind of what to do. It wasn't a question. It was just kind of you start here, you introduce it, and this is how the body should respond. And for most individuals, it did respond that way.
When researchers like you maybe come across something so different or novel in terms of your familiarity with it,
Mhm.
like coronavirus, it just happened suddenly, right? What exactly is your approach towards studying something so volatile in nature while knowing just how much averse the entire human populace could be if you aren't able to decipher how to cure it?
Yeah, I think that put all of us in an interesting position of trying to understand. what is this virus? What's it doing? What's happening? Because a lot of the top scientific officials didn't even know how to respond. Um, so approaching it was the way I approach anything I'm not familiar with, which is just break it down really into its core components and try to understand each of those. We knew coronavirus, it's a virus. Okay, so let's read a little bit about viruses. How do they work? What do they do? What makes this one so special? Why is it called COVID-19? Things like that. Um, I will say I have experience with this. I went into my PhD program having barely any genetics background. They said you didn't need it, and I believed them. And I will tell you that was not the best move. I survived, but I think it was challenging. So every single day, my first year of graduate school, I would come home with a list this long, and I would just sit on YouTube and try to teach myself these materials. So the benefit of living in 2025 is anything you want is available to you. We don't lack any kind of knowledge or data. You can find a YouTube video or just an article on the most niche subject you want. So I think rather than trying to just get all the information that you possibly can, I think now the focus really is on understanding, deciphering this data, and then acknowledging whether it's from a trustworthy source. Do you believe it? How many times has this been validated? Things like that. So I think there is just a plethora of resources for anyone who's interested in learning any topic, honestly. Whether, you have TikTok, look up a short. I guarantee there's someone talking about it. um, you know, YouTube, you just look it up. I know that there are classes. There are a lot of offerings from Harvard, MIT, etc., that will teach you anything you want to know. So yeah, I approached COVID that way, basically, just learning about it. I'm not an immunologist by training or anything like that, so I really did have to take a step back and just try to understand what was happening, and then not only understand what was happening, but being able to communicate that to other individuals. So I always say, if I can't explain it to my mom in a way she understands, then how am I expected to explain it to anyone? Even scientists, you have so much information in your mind about one particular topic, but that doesn't mean that you know anything else about other topics. So I think the best thing we can do is not assume. Even if someone has a PhD title or a doctor, they might not know. They might not have expertise on everything, and that makes perfect sense. Why would they? The data is available. It's the understanding, I think, that is the hard part to train yourself on, but it's definitely possible.
Does simulation, like one approach is doing all the tests, doing all the runs physically, the wet lab approach, sort of, right, doing all those things. The other could be maybe plugging in a computer program and having it simulate a gazillion different permutations and combinations. I mean,
Hmm.
how does it all work? Because I'm pretty sure that both of those approaches are required in tandem to some degree or the other, right? But how exactly, from your end, while you are in the lab, how are you managing the workflow between both these approaches?
So I don't do a lot of this unique research myself, but I do work with other, researchers who do conduct these types of experiments. I think something to mention, I talked over and over again about DNA, RNA, and protein. It's all happening all at once. So even if you're interested in a particular gene, it's usually co-expressed with several other genes. It's never just alone, isolated, lit up. Like, here, I'm the cancer gene. It's very rarely like that. So I think disease modeling using kind of computational approaches is crucial. Um, so sorry, yeah, it's just comparing whether it's genetic sequences, mutations, things like that, biomarkers, and then trying to simulate, if this is expressed here, where else would it be expressed, or what other item might be as well expressed as well. And for that, we need really advanced computational, power. We need everything's cloud computing now, but we need an understanding to simulate, and then from there, kind of narrow it down. So these quantum simulations can model anything from how patients metabolize drugs to which genes are co-expressed, as I had mentioned, in a gene network. Which ones you target this? How does that affect something else? And these data sets that we work with are huge. They're huge. There's 20 million rows in Excel. If you tried to open it, your computer would explode. So it's not like someone can just go down and do a quick statistical assessment not using an advanced computer. So yeah, using these computers, there are ways to predict protein structures, gene co-expressions, things like that, protein gene expression, anything, honestly. If you're looking for drug discovery, you can simulate how the proteins fold and where a drug would bind. Usually you have multiple sites that are predicted, and from there you can kind of narrow it down using these computational algorithms, and then take that information and go test it in a lab instead of having to test 100 different sites. Now it's narrowed down to maybe 7. So from there, you can actually do like loss of function, things like that, just testing to actually see which result in something interesting, which have an effect. And then again, you can take that and feed it back into the computer to refine for future use. The problem is a lot of these are isolated, right? It's usually like one laboratory doing something like this. Of course, they publish papers on it, but this leads me back to just there's so much data, and not a lot of it is aligned. Not a lot of it is in the same formats. There's no consensus on which algorithms to use, which ones are okay, which ones are approved by government institutes. So that's where it gets a little bit complicated. But gosh, there are just so many things you can do with really advanced computers and tools. And like I said, they work in conjunction with the lab. You need researchers in the laboratory space to actually test what the computer's telling you. A simulation alone is only so useful. I know people are, a lot of times protest animal modeling and things like that, which understandable. No one wants to have to deal with an experiment with 500 mice, but until we can actually have really advanced and consistent models in toxicology in particular. I know that they're trying to develop a lot of models on the computer so that they don't have to go through animal testing. But each of those needs to be validated, and then I think in the next 10 years we'll be moving a lot of this testing now just using the computers basically instead of actually in the laboratory.
I'd also think that maybe the very reason why, as you said, personalized medicine is maybe good and more efficient to be catered to, tailored to for one person as in comparison to an entire populace. I think the same could be said true for this. Running a simulation for a particular environmental factors and in general factors associated could be way more efficient, but when you take into consideration all the different things that can arise with variation to the specific whatever genetic
Mhm.
code, tissue structure, whatever it is, I
Yeah.
think it might really help to actually go out and then test it out again, trying to maybe strike that balance between the two approaches, right?
Of course, of course. I mean, I worked a lot in cell culture. So
Hmm.
even just cell culture, I mean, you do the same experiment on 2 different days of the week and you'll get vastly different results. Why? I don't know. You might have eaten lunch before one experiment and not after the other experiment. It could be morning versus nighttime. So there are a lot of factors that play a role that we're not even accounting for. So I think, like you said, just a small scale kind of assessment and validation is useful because once we get that really, we're confident in that analysis, then we can expand by adding other factors and other items that we notice might affect the results. But it really, I think, will be a challenge to take that to a bigger setting with many different data sets with lots of different factors contributing to varying results. I think where we're going to be seeing a lot of future work is actually in probably imaging processing. So I know that they're using a lot of quantum machine learning algorithms to improve resolution, accuracy, and things like that of like CT scans, PET, things like that. And those seem to work really well with large data sets because the more data you feed it, the more you can kind of train it on what's already established. And that currently is being used, and it saves medical professionals a lot of time from having to parse through everything and manually highlight areas of interest. So of course you still need an expert to look over and make sure everything is correct, but I think we'll be seeing more and more of that every day in hospitals moving forward. I think we still have a few years, but I think that's where we're going to really see a huge difference.
Two different tangents. Maybe the first one being the, since we talked about it a lot, a little about the environmental factors affecting, or the temporal factors affecting the nature of the said genetic material, right? The genetic
Mhm.
expression, so to speak. What role would then epigenetics play in the said matter of bioenhancement that we were talking about a few minutes back? Like, how exactly, firstly, how exactly does the environment play a role in maybe a having a different genetic expression while making sure that the genetic code is similar? Like, how
Mhm.
does it all work? And then part two being, how exactly do you work with it to drive towards a situation where you can actually felicitate facilitate bioenhancements or genetic modifications?
Yeah, that's a fantastic question. So let's see. I like to think of genetic material as kind of just this tangled ball of yarn that is in every cell. And so you were mentioning epigenetics, and what that means is just outside influences to our genome. So these things can be like methylation events, where certain parts of the genome are available but silenced, kind of. They can be acetylation, where it opens up. And so, just for people who don't have much of a background, when something is methylated, it's usually silenced because the equipment that comes and translates that portion of the genome, can't really get in there. So what you want to look for is acetylation in open areas of the DNA that can come and get transcribed into RNA and then translated into proteins. And this ball of yarn, as I had mentioned, just constantly shifting in your cells. So it looks very different in the morning versus at night. And there are many influences to why that is. You have an internal clock that can be part of it. Um, what you eat can be part of it. The air you're breathing all affects these things. It all changes the methylation. It changes acetylation. It changes the expressed chromatin or open chromatin. In particular, I mentioned I studied microRNAs a lot. These are small non-coding RNAs, which are really fantastic because they're so responsive to environmental stimulus. So these are RNAs that go bind to an area of RNA and stop it from being translated into a protein. And these in particular, I'm talking about just because they're so responsive. We've found differential expression of these microRNAs based on, again, the air you're breathing, what kind of foods you've had, what time of day it is will change it, which cell you're looking at in particular. So that's just one example of just a piece of the genetic puzzle that changes depending on the time of day, like you had said. So it's really important to take these things into consideration, that it's just constantly shifting, constantly changing. So what that means is that we do need to somehow find a way to do many simulations to understand protein folding is one example, but just actual chromatin accessibility is another. When we do experiments in the laboratory, we do multiple time points a lot of the time so that we have kind of a picture built out of how everything is moving, how everything's interacting. We have also advanced techniques where you can actually map which parts of the chromatin are interacting with other parts of the chromatin, how it's folding, and what types of items might be influencing that. Maybe you treated cells with some agent, or you're testing, ozone or something like that on the cells, and then you can measure this chromatin change. So there are many ways we do this in the laboratory, but again, that's really small scale. So I think we will need, I don't know, just some crazy advanced quantum computing probably to measure, um, again, like folding, structure changes, things like that, because it can coexist at the same time, there's not one answer, I guess is what I'm trying to say. It can be multiple at once, depending on the cell that you're looking at within a tissue. It can be even the cells aren't always exactly the same, even if they're right next to each other. They're all on their own kind of time cycle. So understanding this timing, understanding how the timing affects the structural changes, I think we definitely need the data for, and we need to feed that into an advanced computer. From there, once we actually make understanding of it, we can actually take that and try to figure out, okay, so this area is open at this time of day, for instance. You're targeting that because a drug binds to this area. So at this time of day, you'll give the drug to someone. And they already do that in hospitals, especially with oncology. Certain people are more susceptible to the medicines early in the morning versus at night. So I think, yeah, it's really important to study this. I don't know the best way to do that long term. I think you're not going to see it in hospitals for a long time or anything like that, but just understanding it in a laboratory setting with just a really advanced computer, I think, is the first step. And we're getting there slowly. It's very specific, though, to cell type in a specific instance where you've exposed the cells to something in particular. I think that's where most of the studies are still at this moment. But yeah, I think you'll see a lot more of these chromatin accessibility studies in the future as well, because they all rely on really efficient computational machines. And as we improve in that area, you'll see a lot more of these types of studies. Previously, you couldn't do them large scale because they were just so computationally expensive. Um, but as we get more efficient, I think you'll see a lot of like 3C, 4C type studies, and that stands for chromatin. And you'll see a lot of studies exactly just kind of looking at how the chromatin folds and how that leads to like downstream consequences, what that means for protein expression and things like that.
What about the hereditary diseases? Like, there are some things that get passed down the generations. What exactly is the way that the approach or those medical ways that we are using to cater to those diseases or those problems that get passed down through your genetic code?
I mean, I don't know what they're doing in the medical field with those in particular. I think that would be a good candidate for gene targeting and or things like CRISPR. But I think again, it's hugely important to look at the long term consequences of this. We can cure something perhaps, but does that mean it gets fixed in other cells and still passed down? And or if we do alter it, how does that affect the other processes in the body that also rely on this gene or something like that? Usually a gene is involved in many different things, not just one thing. So by altering its function, we might have unintended consequences long term. I don't know what the current approach to that is, aside from things like CRISPR and like targeted gene therapies. But I think understanding again all the different roles played by a single gene is really crucial to understanding its function, whether we can change it, and what that means long term.
Is there a particular way that maybe through the kind of environment that you're surrounding yourself in epigenetics, right? You can maybe ensure to whatever degree you do not, I mean, pass down something that maybe prove of sort of a challenge to your child or your progeny in the upcoming years, like
Hmm.
any genetic ailment or disease.
Yeah, I think this is an interesting question in multiple ways because not only from a genetic standpoint, I know, for instance, certain types of deafness can be cured, but then you have the deaf community arguing that that's not really an impairment, it's just different. It means that they're really good at other things. So you get bigger kind of conceptual debates that arise over whether something should be removed or fixed, quote unquote, or not. If it's harmless, essentially, again, it's just a diverse population there. It doesn't necessarily mean that they are at risk of anything. So I think that's something to keep in mind, and I think that's something that's going to come up more and more as we look to make these changes, these targeted like gene therapies or alterations, because in the case where someone's not going to survive because they have this gene expression, obviously it does make sense to help. But in other cases, where do we draw the line? We don't want to end up in like a Gattaca type future where everyone's genetically modified, and or honestly it's just going to end up being the rich are genetically modified while everyone else kind of just suffers. There's already debate about whether with in vitro fertilization you should be able to pick the sex. So I can't imagine what it's going to be like if you can actually choose to alter, whether someone will have an ailment or disease or anything like that. Yeah, I sorry lost myself in this here, but I guess the question is, I don't know how they're approaching it necessarily. I think that's where you have genetic counselors are very helpful, because it's kind of a mix between an expert in genomics and genetics and a therapist who can help with the families and talk them through everything. And we've already seen a rise in that kind of job. More and more people are becoming genetic counselors, and I think that will continue as well. And yeah, I think they're probably the best person to ask about something like this, like what the steps are and how it goes to be. But I do think it's fascinating, and think, yeah, we'll see more of these issues arise, I think, in the next several years.
Yes, I think the most prevalent one right now may be trying to the entire subject of age reversal, or maybe slowing down the aging process. And you
Hmm.
see that umpteen times, mostly the rich, they are hell bent upon trying to, maybe just prolong that entire process or slow down to whatever extent that you could. But from my knowledge, there really isn't anything that does not assure you that there won't be any long term detriments towards using all those, right?
Yeah, we've not figured it out yet. I mean, we
Mm.
still don't know why someone can eat this type of food and feel great and another person can't and they feel ill. I think there's a lot of gimmicks in medicine is the problem right now. People have a lot of they get excited, right? It sounds official. It sounds like you know what you're talking about, and you might, and it might be harmless. But I think this is going to continue to be a problem, which is why scientific literacy is super super important, and that's why I try to talk to the next generation and encourage scientific literacy and understanding. Just because someone tells you something doesn't mean it's true. Um, Especially like you said, it's just rich people buying into a lot of things, and there's no actual proof. I guess we'll find out in several years when we see what happens. But as far as I know, there's no magic age reversal anything or drugs or supplements you can take. And even if there were, everyone's bodies respond differently to these anyway. So how can you even guarantee that it would work? I'm still stuck on just trying to get the makers of Advil or whatever to test on women also. Like, that's step one. I mean, forget this longevity, age research, whatever. Even then, it would just end up being like rich white men probably, and that would be a problem.
Mm. Okay, scientific literacy, right? Two things. How do you go on about making yourself scientifically literate? And then maybe while you're on that journey, how do you try to make other people scientifically literate?
Hmm. Good question. For myself, I think the first step is understanding what it is I'm trying to understand. So that could be like going back to the coronavirus example, understanding how a virus works, what a virus is, what makes this particular virus so interesting or unique. And then from there, I approach, with this newfound information, I try to read articles or, look at the latest studies. And I approach it from a place of, I don't know, like disbelief almost. Not disbelief, but just you have to convince me in reading the article. I think I struggled with that early on in graduate school because it's really difficult. If someone prints out the scientific article and they have all these experiments and all these images, and you're like, yeah, sounds good. I don't know. But starting out with the basis of, no, that's not true, and then kind of reading, and they say that this assay proves this. Okay, really looking at the assay, does it prove that, or did they just tell me that it proved it? So it takes practice. It's like anything else. It's very challenging to learn, I think. So that's how I approach scientific literacy, is just constantly reading, especially in areas that I'm not familiar with, and trying to see if I'm convinced or not. And then how I pass along this literacy to others is honestly just through conversation. A lot of times someone will read an article headline or something, or they'll make a statement, and I'll just say, "Oh, you think so?" Or "Why do you think that they did that?" Or "What do you think this means long term?" Just asking questions that encourage critical thinking. It doesn't necessarily need to be scientific literacy. I think literacy and understanding and questioning will naturally make you good at science anyway. You can take that and put it into a scientific context, and now you have scientific literacy. So I think just encouraging people to think critically, especially nowadays, you see a lot of articles, and journalism's not necessarily the best, that say something and then it's a little bit misleading, and or it's not the full story, and or the person who wrote the article is paid directly by the people that they're reporting on. So you really have to pause and just approach it as, I don't believe it. I'm going to read the article and see whether you've convinced me or not. And or do your research and find out like who's writing the article, what organizations are they part of. So I think that's the first step. Again, you can apply that to a scientific context. That's the easy part. I do a lot of mentoring. So I work with Skype a Scientist, Letters to a Pre-Scientist, and things like that, where I get to talk one-on-one or in large groups to students. They can be anywhere from third grade up to high school, and they ask questions like this, like how can we how do we understand science? Is science hard? A lot of times they have this image in their mind that scientists are like these brainiacs who just sit and do math all day long. Nope. A lot of times I get frustrated. I don't understand something. I have to reread. I have to make sure that I'm slowing down to understand. So just encouraging them to do that in everyday life, because if you can get good at it in everyday life, whether you're scrolling through something on social media and you see an article headline, to whatever else, your parent tells you something, you can then apply that to science. So if I notice that someone's particularly interested in a field, whether it's quantum computing or AI or machine learning, I look for a lot of kind of review articles, not research articles, but review articles, which generally summarize a concept. And I often try to send that out to the students, and or kind of just read it amongst my friends. I'll send interesting articles. So that helps me, and I think others, hopefully, kind of at least have an awareness of fields that interest them that they might not have expertise in. It helps build scientific literacy. Um, yeah, it helps build just everyday literacy, I think.
One other very interesting facet, what exactly is the quantum side of biology, bioinformatics, the entire field in
Mhm.
general, the varied places that it can be found or applied?
Yeah, great question. I think it can be applied to a lot of different aspects in biology as a whole. Biology is big, so it spans a lot of different things. I think drug discovery and molecular simulations. So what we talked about earlier, understanding like where a drug would bind, how that's affected by various changes in the environment. That currently is really computationally expensive. Like I said, you can't do it on a classic computer. You just can't. So I know more and more groups, Google, others, Roche, I think, are exploring these quantum approaches to try to answer these questions and really visualize and predict, uh, binding sites. There's also protein folding and structure predictions and things like that, where predictions can lead to understanding disease, because a lot of diseases like Alzheimer's and Parkinson's have like proteins that are misfolded as their underlying cause. So understanding why that causes these changes and how molecules behave as a result is really another area. Um, Personalized medicine and genomics, things like that, being able to compare like large genetic sequences and identify really quickly mutations and biomarkers. Currently, it's an hours long process. That's assuming you have an efficient algorithm that you're using. So understanding not only these genes but the networks of genes, because they all work together and they're all tied together in like this web. So understanding how one change can then affect other things within that same web. I mentioned medical imaging, diagnostics, things like that. Again, using computers to really quickly go through a stack and identify the areas of interest, whether these are like cancerous areas or just areas of the tissue that where the attention needs to be drawn. There's an expression of something. So using these quantum computers can really speed up that process. Let's see, drug optimization, clinical trials. These kind of relate to what I was talking about earlier, but understanding demographic data. So we didn't really touch too much on that yet, but just again, there's differences, right, in demographics and patients. So understanding who might be the best patients for a clinical trial, or why was this so um, successful versus this other drug. So predicting responses and understanding metabolism of certain drugs in certain populations can lead to safer and effective treatments more so than they currently are. And then let's see, disease modeling. So that's, I guess, within epidemiology, which I guess is it's related to biology. It's close. It's all, they're all friends. So these are massive, massive data sets, and they can be infection rates, other genetic factors, environmental conditions at the time of infection. They're just huge. They try to collect as much data as possible because probably they're not going to have another chance. So to study these and make meaningful connections between, this factor was this and this one was this, and it seems like they're somehow connected, even though the average person would not be able to look at that and identify that as a link. So they can help with pandemics, in improving like vaccine distributions. Public health interventions are all algorithmically based. So these are just a few areas I think that quantum computing really will just speed up the process and really take biology to the future, to the next step.
I think again there's this very in multiple different fields right now where
Hmm.
regardless of whatever your particular specialization is, you can actually bleed out into different areas of
Hmm.
study and maybe contribute to whatever extent you can in a particular facet of that particular field. And
Definitely.
that brings me to the question of, you surely do your fair share of programming, right?
Hmm.
uh, for somebody who maybe wants to start out with a field like bioinformatics, or maybe just uh, imbibe the principles of bioinformatics, just work with a biological data set in one of their projects, let's say,
Hmm.
how would you recommend them to go on about doing so? And secondly, what is the tech stack, bare minimum technological know-how that they need, algorithms, programming languages, whatever, to be able to do so?
How would I do that? I mean, they need a computer of some kind. Now you don't even need a very good computer. It's all done on the cloud anyway, so you can connect to servers. AWS comes to mind, things like that to get it done. Google Cloud Council as well, a console. Sorry, you're able to code in. So you don't even need a good laptop anymore. You just need a computer that can connect to the internet. I think in biology, we primarily use R, and I know people look down on R because it's kind of just messy sometimes, but R is heavily used as well as Python and Jupyter notebooks. So if you are very, very, very just a beginner, you have no idea where to start, I think R is a great place because you can have this kind of broken semantics, and you just understand if this one language is not working for you, install a different package, and they use different terminology. It's very easy to work with that. um, Even I work with a lot of clinicians who can barely do anything in Excel, and we try to get them in R because it's just so much easier to look through a data set and parse for the thing that you're interested in. Plus, again, you can reuse the scripts over and over, so that's the beauty of it. As I mentioned, any kind of machine learning or anything like that, you really don't want to necessarily do that in R because you can, it's just really inefficient. So that's where, if you're interested, Python and things like that would come in handy. I'm working with another group here who's done everything in Julia, which is kind of unique, I think, in biology, but like that's great. I'm excited. There's many different ways to do it. So I would start with either R or Python, I think, for the basics. And then if you're interested in going into industry or anything, moving from basic academic biology to more applied, whether it's like in a medical institution or something like that, I would recommend learning workflow languages. So these are ways to connect multiple tools together. You might have a script, but who wants to run the same script over and over and then like, okay, next step, run this other script, next step. Um, these workflow languages are a way to automate the tools so that you can click start, upload your things if they're predefined, and then have your output files at the very end waiting for you. And usually these are done in conjunction with kind of cloud computing and things like that. So it's really efficient and fast. You don't have to sit in front of your computer and constantly be clicking submit to submit jobs or tasks to like an HPC. Um, so there are a million places to learn about these things. Again, just YouTube, TikTok, anywhere they'll just talk about them. So some keywords: AWS, cloud computing, R, bioinformatics. Typing any of those in will give you a wealth of resources that you can go through and find out what is interesting to you. And there are many publicly available data sets to work with as well. So you can search through really anything on the NIH and gain access to a lot of public de-identified data that you can play around with. If you don't have that, you can just use your own spreadsheet. Just make one just to get comfortable playing in R or something, just to work on how to use ggplot, things like that. I think there are a few basic commands that most bioinformaticians know in R, like how to create a box plot, how to do certain things, scatter plots. So really practicing those and getting comfortable with them, and then make it pretty now. Things like that will get you comfortable and allow you to start. I think that would put you ahead of a lot of others just learning these basics as you enter school or the field of bioinformatics.
One final science heavy matter, technical matter, right? The field of the segue towards bio, artificial organs, tissues,
Mhm.
all that. Like, where are we in that regard?
Honestly, I got no clue. I have no idea how far off we are or how anything relates to that necessarily, unfortunately. So I can't even make up something to speak to it.
A close enough thing, then maybe going back to genetics and molecular stuff, right?
Sure.
What is something that people, most people, the general masses, do not really know about molecular biology or genetic biology that actually is a one of the most pressing problems in the scientific field, this particular scientific field?
That is a fantastic question. I'm really trying to think about it. I think one of the biggest problems. Man, my mind is going blank. I think just honestly having I've mentioned it before, but just a consensus on the data. We know that there are labs hard at work producing scientific results. They publish papers, they do things like that. But when you multiply that by millions of different research groups doing it, you get just so much data. And it's like, what do you even do with this? Especially if it doesn't, you can't just combine it into one spreadsheet. So I think that is something they would be surprised to learn, that we have a wealth of data. We have so much data. And I know we're churning out more and more every day, but I think just we need maybe algorithms or some kind of way to catch up to the data, if that makes sense. Like that's really where we need to focus on. I think just making sense of what currently exists. I think people would be surprised to learn that there's very little consensus on how we obtain the data, what types of data. There's no handbook that's like, okay, ask clinical trial patients these questions. It's just different for each company, and or no one sat down and taught me when you're doing, you're working with your cells and doing this. Like, it just depends on what your lab does. Um, So I think that would surprise people. They think of science as just this one thing, but science is just multiple ways to get answers to your questions. And I think we really need to figure out a way then to connect all of these various answers that we've gotten in a way that makes sense.
You speak to a lot of young people, right?
Mhm.
A unanimous advice for all the young folks out there, what would it be?
Just do it if it makes you happy. I mean, I was scared to go into science because it's hard, but it's not the hardest thing I've ever done. That was fine. You'll just, just do it. You're debating whether you want to go to graduate school or, take that test. You're gonna age anyway, so you might as well have a PhD at the very end. Just do it. Stop hesitating. If you are weak in certain areas, there are people who are very nice and they will help you. Like there are always friends in the scientific community. So, believe in yourself, I guess. Stand up for yourself, advocate for yourself, and just do the things that make you happy.
Okay, I think I'm really intrigued by this particular question, but what is the most interesting piece of work that you are currently working on?
Ooh, currently working on at work, I think is. Sorry, I'm trying to think. So, I do a lot of kind of medical imaging assessments. So, we do have some machine learning AI kind of based um, notebooks that we're developing, and I think the idea and the goal is to then take these and apply them to different types of diseases and tissues. So I think that's fascinating to me because I can see a direct kind of outcome. It's not something where it's like, here's a spreadsheet. Like you see the pictures, everything's highlighted, and it tells you exactly what you're doing. So I think that's pretty fascinating. But I work doing so many different projects at once that it's really difficult to pick just one. We have lots of groups that come to us and they need help translating from one workflow language to another, for instance, or they have a paper they published and now they want to take that and turn it into kind of an example study for others to learn how to do this type of assessment. So I love working with a variety of different groups and doing such diverse types of work. So it's really hard to pick like just one particular interesting project. Depends on the day.
I think that is a good place to be in. Surely hectic, but
Yes,
indeed a good
absolutely.
place to be in, where you're doing a plethora of different things that you are really interested in doing.
Yeah.
But okay, the final segment of the show, Dr.
Mhm.
Beck, what would be your word to humanity at large?
Be nice. I know that sounds a little bit vague, but with so much happening right now, just care about each other. Just take the time and pause and think, why is this person reacting in the way they are? And how can I make their day better? I think if we all did a little bit of that, you could still disagree on things, but you would have a lot better of a world to live in.
Dr. Rowan Beck, thank you so very much for doing this conversation. And
Thank you for having me.
it's very very rare that I do not really have an idea of how the time has passed. But I think particularly that was because in this conversation, a lot of my questions that I was really intrigued about, I was just throwing at you randomly. I was all over the place, but you very kindly were just answering all of them. So thank you
I
for
hope my
that.
answers made sense. I tried.
Before